So the next very important terminology of materia medica is pathogenesis. Pathogenesis. Now I have again I'm again repeating this topic, but this terminology I have also introduced you briefly while explaining you the therapeutic method or clinical method. But now in this lecture, I will explain you in detail all about the pathogenesis. So the word pathogenesis is basically derived from the Latin word. One, if you study the Latin word, it is there are two Latin words are combined. One is a pathos and another is a genesis. So pathos means away from physiology, and pathos means something abnormal, something structural which is seen at the level of tissue. And one more terminology, one more part of this terminology is genesis. Genesis is generation. Genesis is, if you take this medicine, it will generate certain pathologies. It will develop certain abnormal changes, and this is pathogenesis. I will just tell you certain similar terminologies. If you can remember pathogenesis is a one terminology very commonly used. Then there is one more terminology which is very commonly used is pathognomonic symptom. I'm again repeating pathognomonic symptom. There is something is called as pathological symptom. Some terminologies like pathological general. So four terminologies I have told you: pathogenesis, pathological general, pathological symptom, and pathognomonic symptom. For the time being, you understand that these four terminologies are altogether different. They have their own meaning, they have their own impact in the study of homeopathic materia medica. For the time being, you just focus on this terminology, pathogenesis. So this pathogenesis means what? Generation of abnormal changes at the level of tissues. That means after administration of a drug to the healthy human being, that drug has capacity to produce certain abnormal changes at the level of tissues or at the level of cell, and this ability of any drug to produce abnormal changes, to produce certain pathologies at the level of tissue, it is termed as pathogenesis. And before going further, we will focus on one more slide emphasizing on the evolution of homeopathic materia medica. I have told, if you can recollect, it is this slide was there in the Science and Philosophy of Homeopathic Materia Medica. Now, how the materia medica is evolved or how the materia medica is constructed. First, the experiments of drug provings are conducted. Then the in charge of that prover does the analysis of all the symptoms collected from the various provers, and in that analysis, he also tries to analyze this drug is able to produce what type of pathogenesis, and then its further clinical information. But this term, there are so three basic steps of evolution of homeopathic materia medica: drug proving, analysis and rearrangement of the symptoms collected from all the prover, and then the clinical confirmation. And this term pathogenesis belongs, or it has its connection with this second step of analysis and rearrangement of the symptoms collected from the drug prover. And it is a kind of analysis. It is a kind of analysis is done by a prover, and then that prover is able to comment upon the pathogenesis ability of any specific drug. So here you remember that this pathogenesis is belonging to the analysis and rearrangement, or basically it belongs to the analysis process after the drug proving. And so it is not the direct information which is collected from the prover. It is an indirected information collected after the analysis of symptoms. Analysis of symptoms collected from the prover, and so therefore this term has a certain limitation. That first limitation is that it is indirect information. It is not the direct information given by the drug prover. It is the indirect information retrospectively collected by the prover after study of the symptom. So this is one of the limitation of this term, that this is whatever information is mentioned in the homeopathic materia medica, it is indirect information. Now there are certain also limitations to this process also. The one limitation is that in the terminology we have understood the pathology ability. Pathogenesis means ability to produce any pathological changes of a drug. But there is a limitation that if we carefully study the drug proving procedures, homeopathic drug provings are not continued till the development of any pathology. So if Arnica montana like drug mentions that it is in the pathogenesis of Arnica montana to have or to produce suppuration pathology, and therefore it can prevent that suppuration pathology. So this information is half truth, because it is not directly collected from the drug prover, because our drug provings were not continued, or the provings of Arnica montana were not continued till the point that the prover develops this pathology of suppuration. So it is a very important limitation of this term. Then whatever information we are getting in the books of materia medica under this term pathogenesis, if it is not directly collected from prover, then from where this information comes? This information comes from the toxicology, from the administration of accidental administration of crude drugs, and it mainly comes from the knowledge of active principles. So atropine, which is a active principle of belladonna, that atropine produces rush of blood towards the specific part. So in belladonna it is mentioned the pathogenesis of belladonna is to produce congestions to the various parts of the body. And why it is mentioned? It was not seen in the prover, but it is the action of the active principle, and then it is mentioned in the belladonna. And then one more important limitation of this term is it is a reverse application of law of simile. Till now you have seen application of law of simile. Now what is this reverse application of law of simile? What does this law of simile suggest? What law of simile means? That drugs are able to cure only those symptoms which they have ability to produce. That means if cinchona is producing fever, cinchona will have ability to produce the symptoms of fever. Have you understood why cinchona is a medicine for fever? Because it is in the drug proving, it has produced symptoms similar to the prover. Symptoms similar to the fever, and therefore it is applicable in the cases of fever. This is a forward, or it is a straightforward application of law of simile. As cinchona produces fever, it has ability to cure the fever. And what will be the reverse application? So the reverse application will be while studying the pathogenesis. The fever Basically, the pathology of fever is what? The pathology of fever is increase in the body temperature, and you know that this body temperature is regulated from your knowledge of physiology. It is regulated from the heat controlling center of hypothalamus. Is it very clear? Fever is, our body temperature is controlled by the heat regulating center present in the hypothalamus. So, in the drug proving of Syncona, it is said it produces symptoms like fever. Okay, it is well accepted. Then now there is a reverse application of this fact. If Syncona produces fever, it must be having certain action on the hypothalamus center, or it must be having action on the temperature controlling center in the hypothalamus, and then it is mentioned in the Syncona pathogenesis. These drugs, Syncona, has a profound effect on the hypothalamus, producing symptoms like that of fever. So remember, it is a reverse application of law of similia. That means first there was a symptom. So if the patient has developed, or sorry, the prover has developed symptoms of throbbing pain with redness and yellowish discoloration of a particular skin, the proving has stopped there. But this throbbing and yellowish discoloration of skin suggests that there is some kind of abscess, and then it is reverse applicable. And then it is reversely considered that if this drug can produce throbbing type of pain and yellowish discoloration of skin, these two are similar to like that of abscess, and then it is said that the pathogenesis of this drug is to produce suppuration. I hope you have understood this reverse application of law of similia. If any difficulty, communicate with me. Then, as this term information collected from the pathogenesis is indirect, it has less value than that of pure symptoms, and therefore it has less value in classical or constitutional prescription. What is classical prescription? It is a prescription which overlooks the pathology. It did not that method of prescription do not bother whether that drug can produce that kind of similar pathology. So, in this method of prescription, classical method or constitutional prescription, there is such kind of information about the pathogenesis will be very less value. So these are, so far we have understood the meaning of pathogenesis, its correlation with the evolution of homeopathic materia medica, and the limitations to the term or limitations to the information mentioned under this terminology in the homeopathic materia medica. Then obviously the question comes, then why, if there are so many limitations of this term, why it is mentioned in our materia medica? And every coin has a two sides, so similarly this pathogenesis has also certain scopes and application, and this pathogenesis terminology has more application in the clinical method or therapeutic method of study of homeopathic materia medica. Remember, there was one important objective of understanding this basic terminologies to correlate study of homeopathic materia medica and that of therapeutics. So this ability of generation of pathology is very important in these two types of method of study of homeopathic materia medica, clinical method and therapeutic method. And it is very good for the various researches which will come or which will come in the futures, that yes, Does the really Syncona-like medicine affects heat regulating center of hyosthalamus? So some kind of hyosthalamus, heat regulating center of hypothalamus. So there has to be conducted certain animal experiments on albino mice or on frog, where the actually Syncona will be given and their hypothalamic center will be studied under the microscope. So it is a good scope for the research. And then very important is that this knowledge has applications even today in certain types of cases. So this knowledge is good to develop low trituration specifics. Now, what is this low trituration specific? I must explain you. Low trituration is what? Usually the decimal scales. And for example, there is one drug which is commonly available in homeopathic pharmacy, commonly prescribed by homeopathic physicians to the cases of hypothyroidism. Now, hypothyroidism means that there is a less production of thyroid hormone because of certain etiologies, and because of that less production of thyroid hormone, certain clinical symptoms develop, and no homeopathic medicine can supplement to this deficiency of thyroid hormone. So there is one important sarcode. You understand sarcode? Sarcode is a medicine prepared from the healthy secretions or the healthy parts of basically various animal. So there is one medicine prepared from the thyroid gland of bull, ULL, or thyroid gland of ox, and that medicine is thyroidinum. And this medicine, thyroidinum, is given in the decimal scale like 6x, 30x, 200x to the patient suffering from hypothyroidism because of this knowledge of pathogenesis. The deficiency of thyroid hormone produces symptoms of hypothyroidism. If you supplement this thyroid deficiency, the symptoms will be under control. So this is the meaning of low trituration specific. So to develop some types of low trituration specific medicines for the treatment of specific condition, this knowledge of pathogenesis is very much useful. Then there are certain schools which has this method of prescription, pathological prescriptions, like Richard Hughes, then William Borick, G.H. Clark, Copperwhite, Cooper, etc. These schools emphasize that you must include the pathology covered in your totality of symptoms. If your prescribed medicine does not have ability to produce similar kind of pathologies, there are very less chances of cure of patient. To make it simplify, I will give you one example. So if you want to treat jaundice, if you want to treat hepatitis with homeopathic medicine, you must prescribe a medicine to that patient which has ability to produce hepatitis in the drug proving or in the various toxicological study of drug, and then only you will get this, you will able to cure that hepatitis. So this kind of thought process has resulted into a pathological prescription school, and that this pathological prescription school uses rampantly, or this pathological prescription school is based mainly upon this terminology, the pathogenesis. The more you thoroughly have the knowledge of pathogenesis, you will be very good physician in this pathological type of prescribing. And why this pathological prescribing is required? Now, to manage certain pathological cases, it is really helpful. So if you are treating hepatitis where the bilirubin level is beyond 20-25, the patient is in a hepatic encephalopathy, you must be able to bring down the level of that bilirubin, and at that time you have to prescribe such medicine depending upon the knowledge of pathogenesis. For example, hepatitis, there are good remedies like Chelidonium mother tincture, Berberis vulgaris mother tincture, medicines like nitram, sulf. All these are the specific medicines for hepatitis, and even the physicians are prescribing whenever they see hepatitis, they prescribe any one of these drugs. So this is the application or the scope for this knowledge of materia medica, which is given under the terminologies pathogenesis. So to conclude, I will say one must study the pathogenesis. However, it is less important because it has its application in the pathological prescription, it has its application in the pathological cases, and that is the reason why in the initial lectures of different ways of study of homeopathic materia medica, I have mentioned all these ways are important. It is not only one way you should always adhere to, you should always follow the study of materia medica in only one way, but you have to follow all the ways to get more and more knowledge of the homeopathic materia medica, and this terminology, if you study carefully the pathogenesis of various remedies, it will definitely fulfill the second objective of this lecture. It will help you to apply knowledge of materia medica in your patients.